I'm Hylane. I'm a PhD student at the University of Brazil. I'm supervised by Professor Wagner Fontes. My work is about the proteomic profiling of neutrophils during a respiratory burst and net formation. It's known that neutrophils stimulated by PMA release nets dependent of NADPH oxidase. Therefore, the early phase of net formation is important to subsequent phases. So, we study the interplay between ROS and net formation by inhibitions NADPH oxidase before and after PMA is stimulated in the early phase for shower, and evaluated the neutrophils proteome, nets, and ROS formation. Human neutrophils were isolated and separated in 5 groups: quiescent, Q, neutrophils stimulated by PMA, P, neutrophil exposed to inhibition, Apassinine, neutrophils exposed to PMA and the inhibition, either after or before the addition of PMA groups PI and IPI, respectively, to verify the possible prevention and reversions of the inhibitory process. We evaluated the kinetics of ROS productions by electron paramagnetic resonance EPR, the chemotaxis kinetics, and the proteomic profile of the neutrophils to examine the possible pathways involved in the first hour of net formation. We showed that the initial 20 minutes of ROS productions is characterized by a high rate in neutrophils stimulated by PMA and PMA stimulated after inhibition. The group PI compared to CRI or IP. Therefore, we identified that this production was enough to promote differentiations in proteomic remodeling. Our analysis showed that the proteomes CAM2 and LIMS1 were regulated by inhibition. PI downregulates compared to P, while IP downregulates compared to P and PI. CAM2 has been cited to participate in net formation. Moreover, both proteins participate in actin dynamic that contribute to migration of the cells. Our preliminary results show that IP group to have an increased number of migration cells compared to other groups. Inhibition of NADPH oxidase before stimulus prevents net productions, whereas inhibition after stimulus does not decrease nets. Thus, we showed that the initial peak of ROS productions is enough to trigger nets formation that apparently occurs through the regulation of the cytoskeletal dynamics. This dysregulation impairs actin dynamic remodeling and net formation in the IP groups. I would like to thank my research group and the Luiz collaboration and the funded agents.